morning, everybody. It's nice to see you here today, and thanks for coming to the second day of the fifth Nabrit conference. We're really happy to have you all here. I want to start by thanking a couple of people. One is Amy Cohen, who has really helped hold things together organizationally and with logistics, and really deserves a big hand. If you see Amy, please give her a thank you. I'd also like to thank the media services staff, and today we have Paul and Laura helping us out. They've really done an amazing job yesterday. Everything was really smooth, and so we should also thank Alex and Connor, who were here yesterday. I think we may see them again this afternoon. And I want to especially thank all the people who asked questions yesterday and encourage you to keep it up. Keep up the good work. Then a little business announcement. If you're presenting a poster, please do have it hung up by 10:45. We're going to start the poster session around then. There are pins out there. The poster boards are numbered. There are a few copies of the directory. If you've forgotten which poster number you are, there's a directory there that you can consult to figure out what your poster number is. The QR code that's here on the banner or on the program will take you to the meeting website, and there's also an abstract book posted on the website, so please feel free to use that. I should, for anybody who didn't hear this yesterday, unfortunately the printed program looks nice but is not accurate because we had a couple of speakers who had to cancel because of illness. The schedule that is on the website is accurate. There are no major changes, but if you really want the accurate schedule, look at the one that's on the website. We have a really exciting day planned today. I want to highlight that at lunch, I'm going to encourage undergraduate students and researchers to sort of hang out in this area so that faculty and grad students know where to find you and come and talk to you about your research, graduate programs, all of that kind of thing. So that will be here in the area near the doorway, and very informal. But to help with that effort, it's also really great if everyone wears a name tag, and I'm really happy to see so many of you doing it. I know I hate name tags, but even I'm wearing one today. So please use a name tag so that people know who you are. I think I have said everything that I need to say right now. So with that, I'm going to introduce our morning session chair, who's Dr. Michelle Dawson, who's a faculty member in the MCB department and has been a member of the Nabrit organizing committee for a couple of years now and has really brought great energy and ideas to the conference. So it's great to have her chairing the session.
So our next speaker is a senior postdoc, Christina Roman Vendrell from Marine Biological Laboratory, and she'll be talking about illuminating mechanisms of synaptic dysfunction in Parkinson's.
Hello, thank you. I want to thank the organizers for inviting me to share my work as a postdoc at the Dr. Morgan Lab in the MBL. And before I get started, I want to talk a little bit about Parkinson's disease and specifically the pathology, right? So as you may know, Parkinson's disease is a chronic progressive disease that is characterized by the degeneration of the neurons of the basal ganglia. What I show you here, so a normal neuron will receive the message, right, and then it will release a neurotransmitter, and then you have normal movements. However, in neurons that are affected by Parkinson's disease, they will start having a lot of problems. Some of them will die, and then that's when you have the movement disorders that are characteristics. In addition to motor disorders, there's also cognitive decline. And so there are multiple causes of Parkinson's disease. There's some genetic, this is a low percentage. There's like a 90% that it is I'm blanking on the word, but it just so happens it's not genetic. And one of the proteins that is very important for this process is alpha-synuclein. So it is very known that accumulation of alpha-synuclein, specifically at synapses, has been linked to this neurodegeneration. However, the mechanisms of toxicity at synapses are unclear, and so that is one of the main questions that my lab is trying to answer, right? What are the mechanisms of alpha-synuclein toxicity at synapses? And so you may know that alpha-synuclein is one of the main components of the Lewy bodies. As you can see, this histology. And these Lewy bodies can be found in multiple neurodegenerative diseases, including Parkinson's, also dementia with Lewy bodies, and some forms of Alzheimer's. And it is important to know that alpha-synuclein, it is a protein that is normally found in the brain. However, it is a protein that is very prone to aggregation, and again, for multiple factors will cause it to aggregate, and then when it aggregates, that's when you get all of the pathology. And so we know that alpha-synuclein exists in multiple structural forms, just to add to the complexity of this protein. It is normally found as a native unfolded monomer. And this protein really likes to bind lipids of high curvature, specifically the synaptic vesicles. And when it binds synaptic vesicles, then it adopts this alpha helical structure. And then we also know that this alpha-synuclein can exist in an equilibrium with multimeric forms. And there are, again, studies that have shown that if you disrupt this equilibrium, that can also lead to the aggregation. And when alpha-synuclein aggregates, it will form these oligomeric structures that are very toxic, and then it will continue aggregation until it forms fibrils. And again, we know that there are toxic oligomeric structures, alpha-synuclein, that it will aggregate at the synapse, and then it will just cause all the problems. And so importantly, in dementia with Lewy bodies, there's about 90% of the alpha-synuclein aggregates are located at the synapses. So again, highlighting why we need to focus on understanding how alpha-synuclein affects this part. And importantly, studies suggest that this presynaptic dysfunction is one of the early events in the neurodegenerative diseases. So again, focusing on the synapse. And so a little bit of background on what happens at the synapse. So right when you have a neuron and it is stimulated, what happens is that the synaptic vesicles filled with neurotransmitter would fuse with the membrane, and then would release the neurotransmitter, and then you pass the message. In order to maintain neurotransmission, you have to recycle these vesicles. And one of the many mechanisms of endocytosis that I show here is clathrin-mediated endocytosis. And this, again, multiple stages. You have the clathrin coats, then you have the maturation, and then you have these buds here, and then they fuse up from the plasma membrane, and then you have these vesicles with the clathrin, then iron coated, and then you have the vesicles filled back with neurotransmitter back to the pool. And we know that alpha-synuclein is involved in the process of in the regulation of the neurotransmitter release, but also in the endocytosis. And again, and also it is known that it facilitates basically clathrin-mediated endocytosis. And so one of the main questions, of my work in the Morgan lab is to understand how does excess alpha-synuclein affects vesicle trafficking. And so to do this, we use the lamprey. And the reason why we really like this organism for our studies is, as you can see here, they have in the spinal cord, they have basically giant axons. And then these are all the synapses. And so this very large size of the axon allows us to inject proteins or drugs, antibodies, etc. In this case, we'll inject alpha-synuclein. And so what we do, again, we can do a control injection, as I show here in this diagram. We can really identify an axon, and then what I show here is that we will inject alpha-synuclein with a green dye, just so that we can see the diffusion. And then we will have sort of different concentrations, like a higher concentration, low zone, and then the control that then we can analyze using electron microscopy. And one of the techniques that I've been recently developed in the lab is to be able to see the injected alpha-synuclein using immunofluorescence. So what I show here is in magenta is SV2, a marker of synaptic vesicles, and then in green, that is alpha-synuclein detected with an antibody. And so you can see here that we have a localization of this injected alpha-synuclein to the synaptic vesicle cluster. And then what I want to show you today, it's more about our electron microscopy project. And so again, this is for you to familiarize with the morphology of the synapse. So it has like a big, large synaptic vesicle cluster, and then we can also see really the clathrin codes. We can identify all the stages, which again, very beneficial for our studies. So yes, again, why we like the lamprey. And so before I get into my data, I want to just how this all started. So in a lab, first before I joined, they were testing the monomeric alpha-synuclein. And as I show in this micrograph, you can see in the blue circles is that when you have an excess of monomeric alpha-synuclein, it affects the stage of the encoding, basically, as I show in this model, because now we have this accumulation of these clathrin-coated vesicles. Now, interestingly, when they injected the dimeric alpha-synuclein, now what you see here, it's a stage 3, suggesting that now dimeric alpha-synuclein, instead of affecting the encoding, is affecting mainly the fission. So again, you have the same protein, different conformations, and it affects different stages of the clathrin-mediated endocytosis. And so when I joined the lab, I was really interested in continuing to characterizing different alpha-synuclein species. And in collaboration with Dr. Tim Bartels and Dennis Selko, they developed a protocol where from the human brains, they can purify alpha-synuclein. And what I show you here, this is the fraction that we injected into the lamprey synapses, and it's mainly multimeric alpha-synuclein with a minor component of the monomer. And basically, this is already published, so what we found was that when you have an increased concentration of this brain-derived alpha-synuclein, it really affects the morphology, right? You have a loss of synaptic vesicles, you have an increase in these large vesicles that we call cisterna, so it really affects intracellular trafficking. And what I show you here is that basically what we have is a redistribution of the membrane, right? We have a loss of synaptic vesicles that it is compensated by this larger cisterna, and then the plasma membrane remains unchanged. And so another interesting phenotype that we found was this, is what that we call sort of fusosomes, and they are this sort of like cisterna-like and complex vesicles, some of them with clathrin coats that we see at the active zone. So it was very interesting because we've never seen it with the other alpha-synuclein species. And so basically, so to summarize this part, so we found that the human brain derived that contains multimer and monomers affects intracellulars, but no clathrin-mediated endocytosis, which is what I showed that happens with the monomeric and dimer. But because this is a mixed sample, and I want to know really what's happening with the multimers, again, back to our collaboration with Dr. Tim Bartels, now they provided us with purified multimers. And now I'm able to inject this multimer into the lamprey, and I'm going to share it with you. This is very preliminary data. I'm working on a manuscript for this. And so what you can see, again, is sort of there's a decrease in the number of synaptic vesicles. There are some clathrin-coated structures, but not that many. So mainly what we see is a loss of synaptic vesicles, and we also see some of the larger vesicles. So I'm going to go here. So again, there's a significant decrease in the vesicles, cisterna, and then, but we don't see effects in the plasma membrane or the clathrin coats, even though we see a little bit of changes in the stage 4. And another interesting thing is that now we have a loss of membrane. So where is that membrane growing? We don't know. That's another question that we're sort of looking at. Well, again, very interesting. And basically, this sort of phenotype is at least partially what I show you with the brain derived, which again was mainly the multimer. And as a control, we also injected monomeric. And again, we also see the loss of synaptic vesicles. We see some increase in the number of clathrin coats that I'm going to show you here in the data. So again, loss of synaptic vesicles, cisterna also are larger. There's no change in their size. But now here we have changes in the plasma membrane. And we also have significant changes in the clathrin coats. So again, this monomeric sort of recapitulates what we see with the recombinant alpha-synuclein. And again, we also have a loss of membrane, so another sort of question that we're going to pursue. And so, but another interesting, again, I showed you earlier with the brain-derived these structures. So then I found them again with the monomeric. Not that many with the multimer, but there's definitely more here. So again, sort of an interesting phenotype. It happens at the active zone, and we really don't know what these structures are. And so basically to summarize this, so based on my postdoctoral work, I have shown that this human brain derived, that is a mixed sample of multimers and monomers, it does not affect clathrin-mediated endocytosis, which is interesting. But then we do see effects in the basically endosomal trafficking, which is sort of the route that I want to go to for my independent research. I want to focus on what is happening with endosomal trafficking at the synapse. And so again, I show you that with the monomers, purified monomers, it affects fission and encoding as shown by the clathrin-coated vesicles and also the ones that are attached to the plasma membrane, and then with the multimer, we mainly see that the larger, basically the losses of vesicles and then the larger cisterna, but not that much effects in the clathrin codes. So again, hopefully with this, I sort of convinced you that depending on the structure of alpha-synuclein, it really affects, it interacts differently with the components of the synapse. And again, knowing how complex these diseases are, again, I mentioned Alzheimer's, dementia with Lewy body, Parkinson's disease, they all have a common, alpha-synuclein as a common target, but it acts different in all of these diseases. So in order to find treatment, then we have to know about this. And so a little bit about, again, my future work at my current lab. We're gonna inject then PD amplified fibrils to look at the synaptic vesicle trafficking and also test different drugs to see if they have any effects correcting the synaptic vesicle defects that I showed you. And that is it. I want to thank my lab, Audrey is over there, and yeah, thank you.
So we definitely have time for a few questions, and I just want to say that she didn't mention, but she also has a new K99. And so, question here, and please introduce yourself.
Oh, me? Hi, I'm Moses, and I'm at McLaughlin in Montana. But I guess I was going to ask, so the human alpha-synuclein that you're injecting for that purification, what antibody are you using then?
So for the one that I show, I used the Michael J. Fox R1. Yeah, that's the one that I used.
Gotcha. And then the second question I had is the lamprey has their own alpha-synuclein,
They
right?
have a gamma synuclein.
Huh?
They
Okay.
have a gamma, yeah.
Okay. So I guess that kind of affects my second question, because I was wondering if there's any propagation of their endogenous alpha-synuclein with what you're injecting.
Yeah, that's a very good question. And again, before I joined the lab, they did the experiment where they basically injected an excess of the gamma synuclein, and what they found is a very similar phenotype to the recombinant monomer. But again, there's a very high possibility that the alpha-synuclein that we're injecting is potentially interacting with the gamma. We don't know. Definitely something to keep in mind.
Okay.
Hi.
Please introduce yourself.
Yeah, I'm Sujitra Kamble and I'm from MMI Brown University. So my question is just in continuation, like you use an antibody to inject to the mice. So you inject it into somewhere the brain side or into the peritoneal cavity, or if you means inject it, does it cross the blood-brain barrier?
So again, what I show you, the preparation is that I dissected a spinal cord, so not the brain. And again, as we can inject pretty much anything that we can get out of the pipette into the axons are fairly big. So again, in terms of that, we are sort of bypassing that by injecting them directly into the axon, and then it reaches the synapses.
Thank you.
Question, Brett.
Hi, my name is Letitia Franklin from Penn State University. My question is towards your future directions, and I don't know much about the drugs that are being used to treat Alzheimer's and dementia and things, but my question is, what is the mechanism that you're expecting to target with the drugs? Like, is it to prevent the misfolding, or is it to reduce the amount of alpha-synuclein that's produced? Those are the only two that came to mind.
Yeah, so again, we have different compounds. One is called CLR1, which is basically like a molecular twister, so that will sort of disrupt the potentially the aggregation of alpha-synuclein, at least the binding with each other. And then we also have MPT compounds that potentially they will interact with or disrupt the interaction with the membranes. So again, with alpha-synuclein, you can potentially target the interaction with the membranes, right, the synaptic vesicles, but also the interaction with itself. So those are sort of like the two main compounds that we're going to be testing.
Greg, you can go next.
Okay, thank you. Christina, this is right over here. Yep, Greg Valdez, Brown University. That was beautiful. A lot of work and a lot of questions. So, what struck me was mostly the synaptic vesicles, the loss of them. So, what's your theory, right? There are so many possibilities. Either it's affecting anterograde transport, it's causing aberrant neurotransmission and depleting the synaptic vesicle pool. I mean, you name it, right? So where are you going to go next with that?
So again, I'm going to be looking into the endosomal trafficking, and so what I show here is just a snapshot, right? So we inject, and then we stimulate, and then we fix, and it all happens within like 30 minutes. But after we stimulate, we don't wait. So what I plan to do is just look at different time points just to see what happens next, specifically focusing on endosomal trafficking. Because again, we see these sort of large vesicles that look like endosomes. So I'm going to be doing more electron microscopy and then hopefully to track that. Yeah.
So we have one more question in the back.
Hi, thanks for a great talk. Katie, Summer at Brown. I had two questions. One is, what do you think the connection is between the synaptic phenotype and cell death that ultimately occurs? And two, I've heard that the oligomers are more toxic than the fibrils, and do you think that the multimeric species, is it also as toxic as the oligomers?
Yeah, so let's see. So your first question, sorry, can you repeat it?
The connection between the synaptic phenotype and the ultimate cell death.
Okay, yeah. So definitely, again, so the function of the neurons is just to keep constantly revealing the neurotransmitters. And so again, when you disrupt that, basically they're gonna start dying, but also there's toxicity. So alpha-synuclein will just start this sort of chain of events that will release the mitochondria is involved, so the oxidative species that are released. So it's sort of like the beginning, and then it starts, and then the environment is just for death, just because of the accumulation of protein. So it really affects membrane trafficking. So everything sort of like stops. And then you have a lot of reactive species. So say that's the connection. And then for the toxic oligomerics, yes, that is definitely what we know in the field. But we're still, because there's so many different structures, even with the oligomerics, and depending how each lab purifies them. So again, it's something that is very, very pressing, just to sort of try to clarify. But there's so many different oligomeric species, but definitely the ones that are the beta sheets seem to be the more toxic, even more so than the fibrils. Again, potentially just because maybe the fibrils are sort of like a way of the system to sort of getting all of the excess and take it out, because it's so much, so that the cell then can keep working. But with the smaller oligomers, that just keeps making the cells just worse. Yeah.
Okay, we have to move on now, but thank you for that amazing. So our next speaker is Sorel Oyenkamp Yingma from Brown University, and he's a senior graduate student, and he'll be talking about pollen tube fitness.
Thank you, Michelle. I hope everyone can hear me well, because today I want to talk to you about something very important, because I want to talk to you about your food and how global warming is going to might force us all to go on a diet. And I don't know if you've looked at me, I don't want to go on a diet, so that's why I dedicate my time into figure out ways to prevent that from happening. But today specifically, I want to talk to you about breeding for tomato and overall crops, and how pollen tube fitness was selected in varieties that have been shown to be resistant to high temperature. And this is, first, I hope I don't have to convince you anymore that global warming is a thing. And not only is it a thing, but it's seriously threatening our ability to produce food. And this is just an example of a field study that was done around the Mediterranean Sea, where they just look at how much tomato was produced relative to the mean daily temperature of the growing season. And what they see is that an additional 3 degrees resulted in about 70% reduction in biomass production of the food. And most of the climate models right now are predicting that we're going to get at least an average of 3 degrees, but even probably more than that. So it's very important that we find a way to reverse this trend and or to make it even go higher, assuming that the human population just keeps on increasing as if we had unlimited resources on the planet. So in my lab, we try to understand why this is happening, but more importantly, we focus on studying two different types of crops. The first type of crop are these one that we just talked about, and this is hiding it, but they're called thermosensitive. Okay, meaning that when you increase the temperature, they experience a significant decrease in productivity. But there are also these other varieties that are more resistant to this phenotype, and these varieties are called thermotolerant varieties. And today we're going to follow two different varieties: Tamalipos, which is from a region of Mexico, and Heinz, which is which was developed by the company that also makes your ketchup. So in my lab, like I say, we are interested in what global warming is doing to this at the cellular and molecular level. But more specifically, we're focusing on plant reproduction. Yes, plants do reproduce sexually, and they do have male and female gametes like animals do. And plants that reproduce through their flowers, and in an immature flower, you will have the male gamete, which is the pollen gland developing in this enter cone, and then inside of the ovary, you will have ovules that are developing. And during reproduction, a pollen gland must land on top of this structure called the pistil, germinate the tube all the way to the ovule, and then every fertilized ovule will go on and develop into a seed that we can either eat or sow for the following season. And if you have enough of these events happening, especially in tomato, you will get a fruit that has maximum size. And we've known that there's a relationship between the amount of seed that trees are able to produce and the weight that the resulting fruit has. So in my most of the research on this field has focused on pollen formation and gamete formation in general, and that's because during the life cycle, the plant during reproduction, the plant spend the majority of its time in this phase. It only spend about 24 hours in this pollen tube growth phase, and maybe that's why most of the research on climate and global warming has focused on this. But for me, I was really bothered by the lack of information that we had on this stage, so that's why I decided to focus on that. And today I will try to convince you of three things. First, that this phase that I say lasts about 24 hours is the most critical for when it comes to climate change. I also want to convince you that thermotolerant varieties have been able to improve their pollen tubes. And lastly, we'll try to look at what major pathways they have selected to be able to do that. So to see if the pollen tube growth phase is important, my colleague Kelsey out of Arizona did a really nice experiment where she limited the stress only to the pollen tube growth phase for about 12 hours. And like I said, she looked at Heinz and Tamalipos. And what she noticed is that, yes, indeed, limiting the stress to this stage is able to recapitulate the loss in biomass that we do observe in the field. But more importantly, the variety Tamalipos was more resistant to temperature compared to Heinz. Again, highlighting the fact that it is a thermotolerant variety compared to Heinz that's more like a thermosensitive variety. However, we cannot just replace Heinz with Tamalipos because replacing the two is already decreasing productivity. So instead, as biologists and as molecular biologists, what we try to do is understand why this guy is more resistant at the molecular level so we can improve other important varieties like Heinz. And to do that, we decided to look at seed production. And that's because the number of seed that this plant produces is directly proportional to the number of successful pollen tube growth that happen. And what we notice first is that under normal temperature or room temperature, these varieties produce about the same number of seed. But when you increase the heat, Heinz experience a significant decrease in its seed production, while a variety like Tamalipos is not significantly affected by that increase in temperature. Again, signifying that the pollen tube, again, is very critical when it comes to biomass and seed production at elevated temperature. So These experiments are nice, but something that it doesn't do is address directly the role of the pollen chip, because we have the pollen chip on one hand and the pistil on the other hand. So Kelsey and I decided to divide the work, so divide and conquer. So she's looking at the pistil while me are focused on the pollen and understanding what's happening on the side of the pollen. So to do that, we had to take the pollen out of the pistil and grow them in vitro. And I designed a really nice high throughput phenotypic assay that allowed us to quantify the different state of pollen during its growth in vitro. So basically, all at the beginning of growth, all pollen tube will be in this unresponsive state. But as time goes by, they must germinate the tube and maintain this tube all the way to the ovule. And anything that causes these events to fail, like for example, the grain not properly germinating or the tube not properly getting maintained, will result in pollen tube burst and a loss in biomass. So because something that I haven't mentioned yet is that all pollen tubes are supposed to lyse. All pollen tubes are supposed to die. However, they're not supposed to die until they reach the ovule. So today we're only going to focus on the most important phenotype, which is the inability of the pollen tube to maintain the tube. And just looking at it first at 28, here on the y-axis you have the fraction of grain that properly maintain the tube in the first 2 hours. And at 28 degrees, we don't really see any changes in the variety, except for example, for gold nugget, that's already starting to show some significant differences. However, when we increase the temperature to 37 degrees, we start to see significant decreases in pollen viability and pollen tube viability. However, her thermotolerant variety Tamalipos is still doing better than a thermosensitive variety Hans or thermosensitive VF36. However, this line right here from Gold Nugget is telling us that it's probably doesn't just have to do with the pollen tube, but that there may be many different roads to thermotolerance. So with that in mind, I hope that convinces you that yes, the pollen tube is important and that thermotolerant varieties have been selected. So how about the pathways? So for the rest of the talk, we're going to focus mainly on the cell wall, and the reason why we're going to focus on the cell wall will be clear in a little bit. But just focusing on the cell wall a little bit, it has to resist a lot of pressure coming from the moving cytoplasm and just the overall target pressure coming from inside of the tube. But the cell wall of the pollen tube is divided into two main regions. The first one, which is the tip and the site of active growth. Usually, this is over here. There's a spectrum that we consider to be soft and flexible. That's deposited, and the flexibility of the cell will allow the cytoplasm to push against it and continuously grow. But along the shank, the pollen tube needs to be able to resist pollen tube burst. So there is a reinforcement that happens by the action of this pectin methyltransferase and this callose synthase complex that make the pectin and callose wall much harder and was resistant to this target pressure. And in between them, there is this transition region that we know is the site of pollen tube burst. So we need to study what is happening over here, and more importantly, we're going to focus on what's happening to callose and to the callose cell wall. The reason being that it's been shown for more than 20 years, or for almost 20 years now, that if you degrade callose from the cell wall using this enzyme beta glucanase, it will favor pollen tube germination, but only to a certain level. Because if you pass that level, it will actually start to increase cell burst. And it's also been shown that if you knock out the callus 5, which is the main enzyme in this complex, you may also be able to inhibit pollen tube growth in the species Arabidopsis. And to find out if callose was important in this process, we did an RNA-seq analysis from on pollen tube grown for 6 hours exposed to stress or not, and we also measured the content of callose in the cell wall of these two varieties. And just looking at the expression of the enzymes in this complex, we can already see that Tamalipos has much higher expression of this enzyme compared to Heinz under control. However, temperatures seem to be down-regulating this complex, which means that we should expect to see a decrease in the callose content due to temperature. However, this is basically what we see, because when we measure the content of callose in the cell wall and we plot everything relative to Heinz, we do see that temperature leads to a decrease in the content of callose in the cell wall. However, again, the Malipo seems to be prime in that it has higher callose content from the start, such that even if temperature decreases, it still has levels that are at or above the level of where Heinz was. So basically what that means is that Tamale boss again has primed itself with a much thicker layer so to be more resistant to leakage. And with that in mind, I can conclude with everything that I've done today, which is I hope that I convinced you that the decrease that we observe due to temperature in the field can be replicated by simply limiting the stress to the pollen chip growth phase. And remember that the pollen chip is a single cell, so this single cell can control whether we all go on diet or not, which to me is fascinating. But what we have hope again that I've been able to convince you is that varieties that have been able to adapt to this temperature have done so by ensuring that the pollen tube are more resistant, such that even if temperature causes them to lyse prematurely, they still have enough grains that are able to go and pollinate the ovule. And the way that they're able to resist this lysing is by thickening the cell wall such that they may be primed by the time that temperature is reached. And with that in mind, there's a lot of people that are involved in this work. I'm just a small part of a nationwide, even international, because we have a lab in Europe now. But it's an effort by the National Science Foundation to improve agriculture here in the US. And I'm lucky enough to be part of this group. And basically, all of these guys can be considered to be my PIs, which is a very lucky thing to have. And I also like to thank my funding and again the organizing committee for giving me the chance to showcase my work. It's been years since I've been trying to talk here, so thank you guys for finally giving me a shot.
So we have time for a few questions. In the back with the microphone.
Hi, I'm JP from UNC Chapel Hill. I was curious, when you were doing RNA-seq, did you see upregulation of maybe heat shock factor proteins, for example? I feel like that is something that would be really prominent here, but I just didn't see any.
Yes, just like it was said earlier, 12 minutes is not enough to resume 5 years of graduate school. So I had to make tough choices. But we do know that heat shock factor and heat shock protein chaperones get upregulated, and we do have evidence that that all of them upregulate them because they all need to keep their proteins folded. However, thermotolerant varieties like Tamalipos and another one, Narcano, upregulated way more than what thermosensitive variety Heinz does. So that suggests that they have a better folded proteome compared to thermosensitive varieties. But again, we can talk. I can show you more slides later if you want.
Other questions?
Hi, I'm Jess from Brown. I was wondering if you have any evidence of if these pathways and mechanisms will translate to other types of plants beyond tomatoes.
Yes, so right now, so first of all, I believe there are many roads to thermotolerance. Right? Each variety may have done something different, and I do also think that they translate to other organisms because I also study a different system, Arabidopsis thaliana, and even though we haven't looked specifically at what's happening to callus, we do have evidence of cell wall reorganization in varieties that are known to be thermotolerant. They are shown to have these proteins that are involved in other plant tissues, but to solidify the plasma, to make the plasma membrane more rigid. And some thermotolerant varieties seem to be selecting for those genes.
Gary in the front.
Gary Wessel, Brown University. In many organisms, the gametes themselves are super temperature sensitive. Is there a way that you can evaluate each of the gametes independent of the delivery of the gametes?
What do you mean each of the gametes?
So an egg or the development of a sperm is sometimes super sensitive to temperature.
Yes.
So I'm just wondering if you can evaluate quality of either of those gametes independent of the pollen tube.
Yeah, that's something I haven't thought about because I'm focusing on the pollen tube, but the gamete could definitely be evaluated by just looking at the progeny, how many progeny are being produced. But for those, we'll have to probably set up the stress before we grow them. But I think there are many studies out there that have already done similar things because like I mentioned earlier, like 99% of the research has focused on applying stress to developing pollen instead of growing pollen. Yeah, but I do think that there are definite. I think Rasha is my colleague, a postdoc in our lab, and I think she might have had evidence of the gamete inside of the pollen tubes dividing early. I think it was early, right? When you apply heat stress, because pollen is so pollen grains, inside of them they have either 1 or 2 cells by the time they become mature. And then if they have 1 cell, when they grow, that cell will divide into 2 cells for double fertilization. And what Rasha has showed is that under high temperature, the nuclei tend to divide faster than they do under normal temperature of the gametes. Yeah, but personally, I haven't really focused much on it.
Thank you.
There's one more question, but we need the microphone in the center.
Thanks. I'm Natalie from Brown. I was just wondering, since in one of your slides you had the words turgor pressure, and it made me think about like a lot of how plants deal with like heat tolerance has to do with water retention. Do you have any information, or like are you planning to do anything looking at the interplay between like water retention, turgor pressure, integrity of the pollen tube cell wall, since I guess like turgor pressure would exert pressure on the wall from the inside?
So during pollen tube growth, they have to import a lot of stuff, a lot of their sugar from the pistil or from the media, and they only have enough sugars, for example, to grow for an hour or two. But one of the things that they also have to import is water, but they do regulate their water content by using like plugs, what we call Carlos plugs. So as the pollen tube is growing and extending, it creates these plugs that will kind of regulate the internal volume of the cell. But in terms of water specifically, when we look at the gene ontology of this pollen tube under stress, I think osmotic stress, yes. Osmotic stress is one of the things that come back, but I just haven't had time to explore it intensively. So we are in the same lab. We can talk more later.
Okay, so we'll move on to the next talk. Thank you so much, Sorrel. So the last talk in this session will be from Dr. Heather Bennett, and she's from Trinity College, and she'll be talking about a neuropeptide signaling in C. elegans.
Thank you. So thank you to the organizers for the opportunity to be able to talk about research that's ongoing in my lab at Trinity College. So before I get started, I want to take an opportunity to highlight all of the work that's ongoing in my lab on various different projects by undergraduate students. But today, for the sake of time, I'm going to highlight the research that's being done by Ginger Watzinger. She's a rising junior at Trinity, and she's been working and characterizing the role of a particular neuropeptide and its receptor, NLP40 and ACE2. Some of the reagents and strains were kindly shared with us by Michael Nune and Derek Seaberth. So while I don't have time to talk about all of the research, I encourage you, please, 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 talk to me and ask me about the other projects going on in my lab at coffee hour and over lunch. So oxygen is really important for aerobic cellular respiration, and there are consequences when there's not enough oxygen. So in the context of humans and human health, the brain is particularly sensitive to oxygen in that the brain comprises 2% of the total body weight, but consumes 20% of the total oxygen. And the consequences can be seen in both adults, but also in the developing brain. And so this is further demonstrated in these MRI images. Here you see an MRI image of an infant that underwent a hypoxic event in utero, and the white represents cellular damage. Now, while you can see here that the whole brain is affected, there are particular areas that are extremely sensitive and vulnerable to this type of stress, and other areas that are a little bit more resistant. There are lots of questions that arise. For example, What are the mechanisms that lead to hypoxic resistance and susceptibility? What are the overall mechanisms that allow animals to mount a cellular response? These questions are challenging to address in higher complex organisms, and so we turn to more genetically tractable organisms to answer these questions. And in my lab, we're using C. elegans as a model organism to understand how these animals mount a cellular response. Now, C. elegans have an amazing ability to be able to sense and to respond to oxygen concentrations. And there are lots of features that make C. elegans a great model organism. And I've listed some of those here. So there's highly conserved genes, the behaviors are well described, and there's 302 neurons all mapped and identified. C. elegans have distinct molecular mechanisms and neuronal mechanisms to be able to sense changes in oxygen. The preferred range of oxygen concentration resides between 5 and 10% oxygen. And when oxygen rises above the preferred range, there's a molecular and cellular response, and distinct neurons become active. When concentration falls below the preferred range, we see activation of the bag neurons. Activation is dependent on the expression of soluble guanylate cyclases, and C. elegans have this incredible ability to be able to respond to hypoxic conditions. We define this as approximately 1% oxygen available, and anoxic conditions. This is 0% oxygen availability. The mechanisms to survive these insults are semi-independent in that HIF is considered to be the principal regulator that allows animals to be able to mount a response to hypoxia. However, HIF is not involved in surviving anoxia. And in my lab, we're interested in understanding the cellular, molecular, and neural underpinnings of animals responding to anoxia. So when there's a change in oxygen, we see that there are changes in cell stress pathways and metabolic pathways. And the regulation is seen in both neuronal and non-neuronal cells. We also see that in C. elegans that there's a behavioral response. And the behavioral response is categorized as areotaxis behaviors. In that, we see that there's an increase in locomotion activity and an increase in these turning behaviors, reversals and omega turns. And this lasts for about a few minutes or more, followed by a sharp decrease. And so it's proposed that these behaviors are there to serve for animals to be able to escape and to find better conditions. And in my lab, we're focusing primarily at the moment and categorizing these behavioral changes. And we are interested in understanding how are these responses coordinated? Is this driven by the nervous system entirely? And what is the role of non-neuronal cells? So among the many things that regulate response, we see that neuropeptide signaling regulates anoxia. And neuropeptides are like a cross between a hormone and a neurotransmitter. So neuropeptides can be secreted from a given cell, and they can have an impact on an adjacent cell. And they can also signal from a cell and act on a distant cell. And neuropeptides have roles in regulating sleep, the many C. elegans behaviors, learning, stress, and anoxia. And in my postdoctoral lab, a medical fellow went on and found that a loss in neuropeptide processing and secretion genes, UNC31 and AGO3, conferred resistance. So animals were able to survive this insult really well. And then later work by a graduate student did a shelf screen looking at all the neuropeptides, loss of function in these neuropeptides to see if she could find a neuropeptide that had the same phenotype as losing neuropeptide processing and secretion genes. And what we have been able to do from that work is to go on and to characterize in more detail some of the molecular mechanisms as well as the behavioral mechanisms. So before I get started and I introduce you into data, let me tell you how we're doing the experiments. we are working with L4 staged animals. This is the last larval stage before animals become adults. Animals are reared at 20 degrees, always on food, and experiments are done at 20 degrees. We remove oxygen for 48 hours, followed by re-exposing animals to 24 hours of oxygen. And of course, we believe in reproducibility and rigor. And so after 24 hours, we count the animals that have survived this insult. We do our experiments in triplicate, and we do the experiment at least 3 times. So what we have found, consistent with published results, wild-type animals survive about 50% of the time. Losing neuropeptide processing and secretion genes leads to animals surviving this insult. And losing NLP40, this is a particular neuropeptide, and its receptor also confers resistance. So animals survive this insult quite well. And just to highlight, we've tested 2 different alleles in each gene, and both alleles conferred resistance. So the next thing that we wanted to do was go on and to ask, where is NLP40 and X2 working in order to be able to regulate the anoxia response? We decided to first work on the receptor, and work out of Michael Nonet's lab showed that X2 is expressed in many places, including in neurons and non-neuronal cells, so the intestine and the muscle. So we went on to ask, what if we rescued X2 in these cells? Would we suppress the increase in anoxia survival? We first tested restoring X2 under its own endogenous promoter, and we found that indeed we suppress the response, the increase in survival. So this told us it was specific to losing loss of function in X2. We did tissue-specific rescue experiments, and we went on to look at the intestine, the body wall, and GABAergic neurons, but we failed to suppress the increase in survival benefit. So X2 is not working in any of these cells to mediate the response. We then looked at subclasses of neurons, cholinergic and glutamatergic neurons. These were particularly interesting because the principal oxygen sensing neurons are glutamatergic, shown in blue, and cholinergics, shown in yellow. NLP40 and X2 have RNA transcripts in these neurons, and so we wanted to test restoration of X2 into these particular populations. We found, interestingly, that when we rescued X2 in glutamatergic or cholinergic neurons, that we found that we suppressed the increase in anoxia survival. So it appears as though X2 was working in these populations of cells. So the next thing we wanted to do was look at the behavioral response. The overall mechanisms that allow animals, particularly loss of function in NLP40 and X2 animals to survive this insult is unknown, right? And has never been characterized. And so what we wanted to do was first test the areotaxis behavioral responses. We looked at forward locomotions and reversal omega turns. And so what Ginger did was place animals into these wells, started the assay, and then counted forward locomotion body bends. And what we found is that consistent wild-type animals increase their locomotor activity, right, as you can see, over time in the beginning stages of anoxia. However, loss of function in NLP40 animals and X2 loss of function animals fail to increase their locomotion. And so then Ginger went on to look at omega turns. This is, again, the animals backing up and turning on themselves, and found consistent with published results that wild-type animals had an increase in omega turns. However, NLP40 loss of function animals fail to have these increase in omega turns. And for the sake of time, I won't show you the X2 data, but it is consistent with what we see, that there's a loss in the omega turn frequency. So this says that loss of function in NLP40 and X2 is altering the behavioral responses. So where we are going to go next, we're going to categorize where NLP40 and AIX2 are functioning for these overall response to anoxia. It appears to be that there's a neuronal role. And so what we want to do is to categorize that and really investigate that further. It looks like glutamatergic and cholinergic neurons are playing a role here, but what we would like to do is really narrow down to particular cells. And so we're going to take advantage of the Krelok system and do cell-specific rescue and knockdown experiments. And we're also going to investigate the mechanism, the behavioral responses in more detail. And so what we would plan to do is to alter NLP40 and AIX2 gene function, so overexpress NLP40 and AIX2 to see if we can alter the behavioral responses in the opposite way, either increasing to wild type levels or increasing past wild type levels. And then look to see, can we alter survival? So collectively, these experiments and these goals will help us understand the biologically relevant cells, which is currently not fully understood, and the overall mechanism that's really enabling animals to survive this particular type of insult, but also sheds light on the shift between normoxic regular oxygen concentrations and the shift to anoxic conditions. And so these are the goals for our for the lab over the next few months. And with that, I will be happy to take your questions.
So we have time for a few questions.
Yes. Wait for the microphone.
Hello? Hello? Hi. I'm Vanessa from Yale University. So you mentioned that you used larval stage 4 worms. So, on the one hand, it seems like it's kind of good that these mutants are able to survive without oxygen, but are there then defects in, progeny, for example, eggs, or the change to adulthood?
So it's interesting that you mentioned that, right? So we see, even in wild type, right, like animals that survive, you look at the plates of the surviving animals, right? There are some animals you would never know that they went through this insult. They look absolutely phenotypically normal. They look just like animals that have never went through the insult, right? And then there's some animals that they clearly look like something happened, right? Like they're smaller, they're paler in color, and so we see this variation. There were experiments done by a grad student in my postdoc lab that tried to parse out these phenotypes and bin animals into different categories and ask, well, will they survive better if they don't look like anything happened? Will the animals who survived but clearly look like they're affected, are there going to be these big overall changes in terms of progeny, in terms of lifespan, but could not find any big differences or significant differences? And then she also went on to look, what about the progeny of those animals? Are they resistant, and couldn't find any significance between the two populations? Carrie?
Gary, the microphone is coming.
Thank you for that. Is there any correlation between survival and the behavior with the glycolytic activity that might kick in those animals?
So we haven't looked at glycolytic activity. We think that there clearly is either they're working in tandem, right? Or something, or one might be happening over the other, right? Something like the behavior changes and then the glycolytic activity. We haven't gone on to look at that.
Can you manipulate glycolysis very well?
So we can manipulate genetically with loss of function or gain of function mutants. I believe Daniel Colin Ramos has some biosensors that he's working with that could be of interest, but we haven't gotten to that point yet.
Very good. Thank you.
Okay, are there any other questions? Well, there's no more burning questions, we'll stop here. Well, thank you very much for a great talk.